Hello everyone. In this video, we will discuss a comprehensive tool that has been tremendously used in past years for differential expression analysis and pathway analysis. The tool is basically a very interactive tool for biologists and early bioinformaticians who had no idea how to perform this analysis because they had no knowledge of coding, and secondly, they could not understand the terminologies used in databases, and thirdly, as the information is very scattered in different databases, so there is need to follow a particular workflow to link these tools, and that is little difficult for biologists to be. So this tool is basically called the iDEP tool, that is integrated differential expression analysis and pathway analysis tool. So the authors of this research paper are Steven, Boson and Ronan, and this paper is published on 19 December 2018 in BMC Bioinformatics. So you can basically download this PDF over there, and you can highlight the important parts, and this research paper has 118k accesses and 1364 citations. Now let's move towards the abstract part, like what is the aim of this tool, why they had basically made this tool, because already there are thousands of other tools that have been present to perform these type of analysis on RNA-Seq data. So how they got the idea of making this tool. So basically they had mentioned in the abstract page in the background section over there, so basically they want to reduce the time for analysis, especially for biologists and early bioinformaticians who are basically just stepping into this field and could not understand terminologies used in databases. And there are various biologists who can perform their wet lab work very easily, but they could not perform their work in dry lab, and for some biologists it is very horrible to perform their analysis over there, and they hesitate for it. So basically iDEP has removed all of these hesitations for biologists, and they provide a very interactive and user-friendly platform for biologists. So basically they say that the other tools are very time-consuming and challenging, so they made this tool to save time and to make it easy for users, and it is a user-friendly tool for data analysis, differential expression and pathway analysis. In the result part, they had basically mentioned that they had used 63 bioconductor packages, 2 web services, and they basically shows 220 plants and animal species over there. And secondly, they shows that they had basically verified their results by using 2 datasets. First dataset was based on the lung fibroblast with HOXA1 knockdown, and secondly they worked on mouse B cells, where they find out the functionality of functional P53. What is the role of P53 presence or absence in mouse B cells? From both these examples, they validated their molecular pathways and they generated novel testable hypotheses. This is the link from where you can access their tool. You can click on this link and click open link in new tab. So here in the background section that they had mentioned why they had made this tool, that I had already mentioned, they want to solve the problem of biologists. And so here in this tool they perform the quality control, pre-processing, mapping and summarizing the raw sequencing reads. And here they had mentioned that they use the R programming language for visualization and statistical analysis, and they use different packages like DSeq2 for differentially expressed genes identification. In the last para of the background section, they had mentioned what are the tools that were already present and how this tool called iDEP is different from these tools. They mentioned StartApp and Biggest tool and Sloth tool, Shiny NGS, Diva, RNA-Seq, etc. So you can read them and explore them one by one by basically visiting these links. Here they had mentioned how their tool is different from these tools in these 5 advantages. First of all, they shows that they provide the automatic gene ID conversion with broad coverage. And second functionality that they had provide is they provide the species of both plants and animals for pathway database and gene annotation. And thirdly, they provide the in-depth EDA and pathway analysis. And fourthly, they provide the web services you can use the APIs of KEGG and SpringDB for pathway pathway interaction and network interactions. And fifthly, they provide the R script, like if you want to use your own data set in their tool, you can simply upload it and can perform analysis of your own choice. Basically use the tools called Gene Pattern, Galaxy, and Cybers. If you want to know how to perform RNA-Seq analysis in Galaxy, you can basically watch my tutorial where I had made a video on how to perform RNA-Seq analysis on Galaxy server. Basically, it's a very amazing tool, also for beginners who don't know how to code. Then they basically validated their findings of this tool by two datasets. Firstly, the first dataset is focused on HOXA1 knockdown by small interfering RNA in human lung fibroblast, and second dataset is based on the downregulation of cell cycle genes in E2F1 and its target genes, including that has a role in microRNA blocking G1S transition in mouse. This is the workflow that has been used by this tool. You can click on this full size image. First of all, they create the expression matrix, then they remove the IDs that are duplicated, then they apply the filter and they remove those genes that are that has a low expression rate. Then they transform their IDs into the IDs that they want to perform k-mean clustering and to find out their interaction networks using StringDB. After finding out these clusterings and network interactions, they made the clusters of these genes to find out the enrichment analysis using String API. Here they use the Biomart database. The Biomart is basically the part of NCBI, and they use this database to basically map their IDs to ensemble gene IDs, and they convert their IDs to counts data, and then they transform their IDs to perform lemma trend and to generate heat maps, to perform principal component analysis and TSNE. And for this ID conversion, they use PGSCA package. And here they had mentioned all of the other tools that they had used in their tool, like they use the KEGG API to shows the the pathways, and they use Biomart database to show the gene information, etc. Now next, in the result section, they shows that the main functionalities they used is pre-processing. In pre-processing, they had basically filter out the genes that have low replication, low expression rate, or there are some genes that are duplicates, so they remove those genes. Then they perform exploratory data analysis. Then thirdly, they perform differential expression, pathway analysis, and visualization. Now here, for gene ontology, they use these three terminologies that involves biological processes, cellular components, and molecular functions. And this BP involves 15,796 gene sets, and CC involves 1,916. And the source from where they get these information is ensemble 92 version. And for KEGG, they use the KEGG database. For KEGG pathways, they basically use the KEGG database that involves 327 gene sets, and the release of KEGG was 86.1. And so in this way, you can check them one by one, like for which functionality they use which database. Like for drug, they use drug bank, T3DB, CIDR, stage, etc. And total genes that they have used is 1,40,438. Here they had mentioned their goal, they want to make this tool an intuitive, graphical, and robust tool for experienced bioinformaticians. They can also use this tool, but specifically it has been made for biologists and early bioinformaticians. So in use case one, basically they had explained in detail about both use cases, but in this video we will only understand the use case one because we only want to get an idea of how many genes that they have used, and we will take a look on the graphs that they had generated. So in the first case study, where they worked on the HOXA1 gene, that is homeobox A1 gene. whose that has been knocked down by the presence of small interfering RNA. Now here they find out the effect of this, like if this HOXA1 gene has been whose expression has been reduced or it has been completely shut down. So what is the role of this gene on human lung fibroblast? So in the next section, they in the first step they perform the pre-processing and exploratory data analysis, where they convert their IDs to they perform the default filter or CPM, and they shortlisted 13,819 genes after performing the pre-processing and EDA. Here in this paragraph, they shows that they had used 3 replicates of each of 2 biological samples. One is control sample and other is HOXA1 knockdown samples. So they used 3 samples of HOXA1 and 3 samples of control samples. Here they show the distributions of transform data after performing EDA and pre-processing. And this is the scatter plot, this is the box plot, and it shows that their data is completely normalized because the central line, this median line is completely this median line is in center, so it shows that the data is completely normalized. Then they shows that iDEP also has the capability to show the expression level of 1 or more genes. Here they show the expression level of HOXA1 gene 10, 2, 3, 4, 5, 6 and 7, and it shows that like which gene has been upregulated and which gene has been downregulated. The red color shows upregulation and blue color shows downregulation. Same goes for second case study where they use E2F 1, 2, 3, 4, 5, 6, 7 and 8 types. Here they had shows that they shortlisted 1,000 genes by hierarchical clustering, and after performing the hierarchical clustering, they also confirmed their results by correlation matrix and k-mean clustering. This is the image that they had generated. This red region shows the control samples and this blue region shows the treatment samples. So this control sample shows that these genes are have low expression rate, but in treatment sample basically the expression of these genes has been increased, and here it shows that the red colored genes have a high expression and green colored genes have low expression. Same goes for this PCA where it has 82% variance and PC2 has 6% variance. This circles shows control samples and this triangle shows HOXA1 knockdown treatment samples. They used DSeq2R package to identify these DGs, and there are 907 upregulated genes and 1097 downregulated genes. And the threshold that they has used for false discovery rate, that is less than 0.1, and fold change should be greater than 2. They generated their volcano plots and MA plots to find out their transcriptomic response. They also generated this plot with the help of Plotly, and you can just zoom in and out these images to find out their average expression rate. So here basically this red color shows the upregulation and blue color shows downregulation and black color shows no significant difference in their expression rate. Now, after finding out the upregulated and downregulated genes, they perform the enrichment analysis on these genes to find out which genes are mostly present in which pathways. After that, they perform the GO analysis on them to find out the biological processes, cellular components, and other areas to find out how many genes are involved in how many pathways. So here our downregulated genes are mostly present in these pathways and upregulated genes are present in these pathways along with the number of genes involved in each pathway. So here maximum genes are involved in organelle organization, that involves 350 number of genes, and 45 are the lowest numbers of genes that are involved in mitotic sister chromatid segregation. But here the one problem is like some of the pathways are simply ambiguous, like cell cycle and cell cycle process are both the same terminologies. So in order to basically get rid of this problem, they perform this enriched terms, and they basically summarizes this correlation analysis. And they generated hierarchical clustering trees and network. Like here, this green color shows the down-regulated pathway that basically they perform this correlation again to differentiate the up-regulated genes and down-regulated genes clearly, like how they are, how much their expression is different in each pathway. Now here the green color shows the down-regulated, and red color shows the up-regulated genes. After now they have performed the co-ontology one by one on the biological processes, cellular components, and etc. And for KEGG pathways, they use the KEGG database to find out the upregulated and downregulated genes, and then to find out the interaction network that they had generated here. For this, they use string DB database and they basically use their string API to access their interaction networks. So here, basically, these are the genes that are highly correlated with each other and has high interaction. So you can basically click on this image and can analyze. Now here you may read these paragraphs for your understanding. Now here in pathway analysis, they had mentioned they use a gauge as a method for enrichment analysis and KEGG as a gene set. And here for pathway analysis, they use PathView bioconductor package. Now let's move towards the discussion part where they had mentioned the limitations of this tool. So basically they used a Shiny platform for this network that has its own drawbacks and limitations. And for interactive plots, they use Plotly and ggplot2. And here they had mentioned what are their findings for each of these dataset that they had used for the validation. Now here in the conclusion part, they had basically mentioned they update the information from ensemble every year, and they compile the information for model organisms as in MSigDB and GSKB based on, and also they also prefer the users' request and their feedback, and they also basically want to add more functions by additional bioconductor packages. This is basically the methodology that we had already discussed in the figure 1. You can basically click on this image. This is the image that we had basically discussed earlier. If you have any confusion about this workflow, you can read this methodology for your understanding. These are the abbreviations that are used in this research article. That's it for this video. If you have any question about this research article, or if there is any confusion, you can just drop your question in the comment section and I will definitely try to resolve it. So that's it for this video. Take care. See you in the next video.